All right, well, Stephen, thank you for the invitation to come and speak today. Um, it's actually a privilege to come and talk about some of our new data in this area, which was just published as this project was started initially at Rutgers several years ago and has transitioned here to the NIH. So briefly discuss about hepatitis C virus. We are living through a viral pandemic, and there are other infectious agents out there. Hepatitis C is a particular problem. It's a small enveloped single-stranded RNA positive sense virus, sort of a cousin for SARS-CoV-2. It's in the Flaviviridae family, which includes Zika and dengue, West Nile, other members in that family. It was initially identified in 1989 by Harvey Alter, who's here at NIH, Mike Houghton, the University of Alberta, and Charlie Rice. They won last year's Nobel Prize in Medicine, 2020. Clinical manifestations are, these infections tend to be chronic mostly. You can end up getting cirrhosis, hepatocellular carcinoma. There's about 170, well over 171 million, 70 million to 100 million chronically infected people worldwide. And this is some new data out of the CDC showing the age distribution. And what you notice for this period here is what you notice is you have a biphasic distribution. So the sort of 50s and 60s, these are baby boomers that were infected prior to blood supply being screened and public health issues being implemented, dental work, sterilization, medical procedures, etc. And this second phase right here in this younger cohort is an ongoing problem. This is IV drug use among millennials. And this is a huge problem here in the United States. And also what you're beginning to notice here is this sort of very small but significant blip here at the newborn level. And this is vertical transmission. So if you notice here that this is sort of in the late 20s, early 30s, a lot of people are having children. And this is vertical transmission from the mother to the child. As Guy mentioned, there are numerous drugs that are available, which have a very high cure rate for hepatitis C virus. The downside is they're extremely expensive, $800 to $1,000 per pill over a course of 8 to 12 weeks. And it's one pill once a day. There is no approved vaccine for hepatitis C virus. And the latest clinical trial that just ended about a year or two ago actually showed no protection versus placebo. So no infectious disease has ever been cured by or eradicated by treatment alone without an excellent vaccine. And so this is just an overview of the genome. And today we'll specifically talk about envelope glycoprotein 2. Just some more information about drug overdose and in the US. And what we're seeing now is a new spike. It's been ongoing for a while. This is a lot of heroin use and fentanyl being introduced into the drugs that are out there. And then you see this large spike. This is due to the pandemic. And so this is just from a recent New York Times episode, New York Times publication, and data was from the CDC. And so we're seeing about 90,000, over 90,000 people dying every year from drug overdose in the United States. A lot of that is heroin. And so just on comparison on the side here, peak deaths from gun use, gun deaths, HIV, and car crashes are between 40 to 60,000. So this is a huge spike that we're seeing here in the United States. And I also wanted to highlight that this problem has been ongoing in New Jersey. Ron Nahas is a very well known infectious disease doctor in the state of New Jersey. He's got the largest ID practice in the state, multiple clinics around the state. And Ron contacted me several years ago because he noticed a spike of new HCV cases that really hadn't been seen in the state before. And most of that, again, is through IV drug use. He does work with the Princeton House, which is a psychiatric facility outside of Princeton. They have an opioid detoxification program. And what he noticed between October 2014 and June 2015 is they had 861 unique patients in that time, mostly from Central and South New Jersey. The vast majority, as you see, 66% were of the age of 17 to 35. 43% of those people were HCV antibody positive. Now that is a particularly telling note because a lot of HCV patients do not make antibodies or have very low antibodies. So this means they were either probably an underrepresentation of the number of HCV positive patients. And so Ron tried to enroll these people into some clinical trials using some of the drugs that Guy had showed. The big problem is getting compliance. So they tried to enroll 184 of these patients. 16 actually showed up for their follow-up appointments. 3 were started on these direct-acting antivirals. 2 were thrown off the regimen because they didn't adhere to the 1 pill once a day regimen. 1 was actually able to be cured and then returned to his or her environment and got reinfected when they started to use IV drugs again. So we desperately need a vaccine to sort of treat this population. And so the major determinants of vaccine are going to be either E1 or E2, the envelope glycoprotein 1 or 2. They're small proteins, highly glycosylated, numerous disulfide bonds. We spent a long time at Rutgers figuring out how to make these proteins for both structural studies. They're very challenging to work with, have a lot of disorder in them. E2 is by far the better characterized one. It binds to the human receptors CD81 and scavenger receptor class B1. And so we'll specifically talk about this interaction today. HCV has a very, very complicated entry pathway, more so than a lot of other viruses, definitely more so than like SARS. It has numerous receptors. It goes through this very complicated pathway and eventually ends up in the endosome with CD81. So somehow during this pathway, it's transitioned to CD81 and that is brought into the endosome. And there's a low pH triggering event. So we know low pH during endosome formation and maturation, the pH drops. And that is an important trigger as well as the presence of CD81. And so we were very interested in CD81. It's been known for a long time, and it's been very challenging to get E2 and CD81 to interact and behave properly. Just a bit more background on CD81. It is a tetraspanin family member. It's a small protein. It's expressed on all nucleated cells, so basically anything other than red blood cells. It's part of the B cell receptor complex. Interestingly, it's required for both hepatitis C virus and plasmodium infection of liver cells. And the direct binding of E2 has been shown using a variety of molecular biology techniques to be localized to this large extracellular loop, or LEL. The important thing for vaccine design is that the majority of known HCV neutralizing antibodies block the interaction of E2 to CD81. So that's why we became very interested in this in understanding how E2 engages with CD81. And so we solved a number of crystal structures in this area, 3 particular. We did one many years ago, just E2 alone, which I'll talk about in a minute. We have 3 new crystal structures here. So we have, this is the structure of E2 bound to CD81. E2 is in red, CD81 is in blue. I should mention that this needed to be tamarin CD81. Tamarins are small monkeys, I believe they're technically classified in. They're not susceptible to HCV entry, but the the tamarin CD81 can substitute for the human CD81 for hepatitis C virus entry. And actually it binds substantially better. So that's a really interesting observation that we found. And so here's the complex here bound to non-neutralizing antibody that's needed to stabilize the protein. Here's the same antibody bound to the free form, and then this is the tamarin CD81 structure alone. So we have these three structures here, and this is just the LEL domain. Interestingly, we started to get these crystals of the complex right before I left Rutgers, and when we came here, we got crystals were not the problem. We did notice, though, that all of the crystals were formed at pH 4 to pH 5. So we began to think about this, and late endosomes are around pH 5, maybe a little bit below that. And so we began to think, well, maybe that was necessary, pH was necessary for complex formation. And it actually does stabilize the interaction. We showed that via ITC, which I'm not going to talk about. But basically, we sat around, made the complex, and dropped the pH to about 4.5, just split the difference between what we were noticing. And so that was really the key in order to get crystals that diffracted to around 3.2 angstrom resolution for the complex. And so here's the two E2s that we're showing here. Again, bound to the same antibodies. The antibody's been removed. And what is clearly obvious is that this loop here, which was known to be important for CD81 binding, it's called the CD81 binding loop. What we notice here is this conformational change. So in the low pH form of E2, it's actually very similar to other structures that were done at neutral pH, with this loop in sort of a retracted position. Upon binding CD81 and low pH, we believe this loop undergoes a large extension. And so when I talk about this large extension, I'm showing that here. So we're talking 13 to 15 angstrom movement of this loop. And that is denoted by these highly conserved, absolutely conserved two hydrophobic residues, this tryptophan and this tyrosine. And you can see, again, superimposing it, you can really see the extension of that loop. And so I just wanted to talk a little bit about the mechanism here. We're still sort of working this out. But what you see here is this very N-terminal portion right here that I've highlighted in red. And so that abuts the CD81 binding loop. And upon binding CD81, this sort of longer N-terminal portion here, which is disordered in this structure, actually becomes ordered and sort of wraps around CD81, which I've sort of depicted as a sphere here, but kind of just wraps around CD81. And what it does is it pulls that N-terminal piece down, and moving it down actually allows this loop to spring. Now, when I was at Rutgers, we solved in 2014, we solved the same structure without this N-terminal red part, so just the core domain on its own. And now, when we got back and looked at that structure, actually this loop was disordered initially, so we noticed that. So again, we've removed this N-terminal fragment that kind of stabilizes this loop, and now the internal CD81 binding loop is free and disordered. Again, consistent with the fact that either CD81 binding sort of moves the loop, or you can lose the N-terminus, or you can just delete the N-terminus and the loop is free. So why is this particularly interesting is we then went to the PDB, we downloaded the full-length human CD81 structure that was determined a few years ago, and we superimposed our E2 onto the human structure. And what you see, we had two complexes in an asymmetric unit. We embedded the full-length CD81 into a hypothetical bilayer. What you see is that these two hydrophobic residues are basically within 5 to 10 angstroms of the bilayer. And so if you consider the extra hydrophilic part of the POP-C, which is the bilayer here, these are practically in the membrane. So it would suggest that E2 will actually insert into the membrane. And that information was sort of already out there, that we knew CD81 and low pH could assist the E2 into membrane association. It was sort of a not well regarded, not well understood property to E2, and sort of disregarded. And so we went back and actually tested this in a liposome floatation assay. And basically what we did is we took complex or protein alone, mixed it with lipids at 37 degrees for about an hour, added 1 molar salt to sort of disrupt the non-specific interactions between positively charged protein and the negatively charged liposomes. We then float them by ultracentrifugation. And what we're showing here very, very quickly, very simply in this graph, which is sort of a quantitation of the Western, E2 at pH, neutral pH, or so it shows some binding. You add CD81 at pH 7.5, it's a bit more. You lower the pH and E2 binds better, but the best one is low pH in the presence of CD81. And we believe that really these two are the critical residues. So we've mutated them and the activity is totally dead, worse than what we see at pH 7.5. And so entry of envelope viruses. So this is a big deal, especially in these days. There's 3 well-known classes of viral fusion. Basically, the schematic is shown here. You have a viral membrane. You need to bring that very close to your cellular membrane. They need to interact. There's a conformational change that could either be receptor binding or low pH or some kind of trigger. And there's another conformational change that then brings these 2 membranes together and allows for fusion to occur. And so these are the 3 major classes, class 1, class 2, class 3. SARS and flu are in class 1, as well as HIV. Now, the big problem here is that HCV has never been characterized because we don't really understand how it does this. And so we still don't understand, but we found, I think, the first clue on which protein is responsible for this initial sort of insertion into the human bilayer of the cell. So this is very early in the process of sort of an intermediate, maybe, in the process of how HCV enters. So right now, it's still not classified.
Time to wrap up, Joe.
Yes, and
Thank you.
So this is just showing sort of our schematic. There's probably an initial form and sort of a post form. And then just to leave you with, I think a very, well, a relevant area, right, of structural biology assisting in vaccine design. We are all receiving stabilized forms of COVID spike protein. Those were guided by structural biology. And so we hope that the structure allows us to begin thinking about stabilizing this protein and possibly doing either mRNA or more classic virology or vaccine-based design. And so with that, I want to just end acknowledging the group, my current group. Really want to just emphasize Ashish Kumar's fantastic postdoc who did a lot of the structural biology and biochemistry, as well as 2 Rutgers graduate students, YY Wang and Samantha Yost. Both were students at Rutgers, and so grateful for all their work and efforts over the years. Lastly, just to thank my colleagues, Jeff Cohen and Wei Bu, as well as long-term collaborator, Arash Kakui. So with that, thank you, Stephen. A privilege to talk to you guys about this again.